All right, in the last lesson, we had a look at some of the components of the second line of defense. So we looked at macrophages, neutrophils, mast cells, and dendritic cells. So all of those being cells, leukocytes that are involved in the second line of defense. What we're going to look at today are a few other things. We've got complement proteins and another type of molecule that we look at, as well as a few responses. So if you remember, we drew this flow chart, which is kind of everything that we're going to cover in this topic of the immune response. We've done barriers, we've looked at phagocytes and those other cells in the last lesson. And today we're going to look at this, these other molecules, and these two really important responses that our bodies go through as part of a non-specific response. Okay, so the first molecule that we're going to look at are called interferons. So interferons are a type of cytokine. So if you remember that cytokines are a type of signaling molecule that are involved in the immune system, these are an example of that. So when a cell is infected with a virus, the cell will release interferons. Okay, so we've got a cell here and we have this virus in there. So this virus is going to use that cell to replicate itself as viruses do. Now, the interferons that are released by this cell will be what we call paracrine signals. Now remember we said that signals that act upon themselves, on the own cell, are autocrine, and paracrine signals act upon the cell next to it. So these interferons will move to the next cell and make some changes to that cell to protect that cell from that virus. Okay, so we go back here, we've got here our interferons. So these are those cytokines. They've traveled from one cell, which has a virus in it, to the next cell. And the aim is to protect this cell from the virus. Okay, it's kind of too late for cell number one. The virus is in it, that cell is probably going to be destroyed, but we want to protect the cells around it. Okay, and there's a few ways that that might happen. These interferons might signal to this cell to prevent the virus from entering, or maybe to stop the virus from replicating once it's inside, or actually to stop the virus that has replicated from leaving the cell. Okay, so the important thing is that interferons act upon the surrounding cells. They're almost like a warning or a signal to the cells around that there is a virus, and those cells need to protect themselves. Okay, so just jump into question one after you've taken your notes and answer that. All right, we've got another type of molecule called complement proteins. And this is not complement as in you look lovely today. To complement is to kind of assist something or to work with something. So complement proteins are proteins that are made in the liver and they circulate in the blood in an inactive form. So what that means is they're always circling around in the blood, and then when they're needed, they get switched on. And what actually switches them on or activates them is pathogens. If there are pathogens identified in the blood, these inactive proteins get activated. Once they're activated, they kind of work with each other. So there's about 20 of these different complement proteins in the body, 20 different types, and they'll start working with each other and they create what's called a complement cascade. Okay, you probably remember the term cascade from apoptosis. This is when molecules kind of interact with each other and cause a series of events. Okay, so basically we've got these proteins, there's a pathogen around, they've been switched on. There are three main things that these complement proteins will do or will achieve. Okay. The first thing, well, it's not necessarily in order, but one thing that complement proteins might achieve is called lysis. Now, lysis is when something breaks or bursts. So let's say we've got bacteria. This is our pathogen, okay? Complement proteins might make little holes in these bacteria, so create these little pores, and that will result in the bacteria bursting, which will destroy it. Okay, so that is one thing that complement proteins might do. There's this big fancy word called opsonization, which basically, it says here coats, which is probably easier. That is when the complement proteins will coat the surface of the bacteria or the pathogen, so basically surround it and make it easier for phagocytes to recognize them. Okay, so here we've got these complement proteins surrounding these pathogens so that these phagocytes will come along and consume it. Okay, and the last thing that these complement proteins might do is called chemoattraction. So basically using chemicals to attract, and that is when the complement proteins will basically gather around the pathogen and attract phagocytes to the area once again so that we consume that pathogen. Okay, so all three of these are ways that complement proteins help or complement the other things that are going on in the second line of defense. Okay, so just once you've got those notes written down, jump into question 2. All right, venom inhibitors. These are another type of molecule that basically exist in our blood, and if the body encounters certain types of venom, these venom inhibitors will bind with that. Okay, we don't have these for every type of venom, which is why some venoms can be fatal because we don't have ways of protecting against them. Okay, I don't want to spend too much time on this. I've never seen this come up a lot in the exam, but it is in the textbook. It's worth looking at very briefly. So venom itself contains various toxic proteins that damage cells. So if we look down here, if there's no venom inhibition, what happens? We've got, say, a scorpion, and it produces a type of venom. And in that venom, we have these little proteins. Okay, so they are the toxic proteins. If they go into your body, let's look at the cell now here. What these toxic proteins have done is they have blocked a channel protein on a cell surface. And by doing that, it means that certain things can't get in or outside of the cell. Okay, that would be an example of how a venom can damage cell function. And this might look quite minor, but if this is happening in important cells, this can cause paralysis, and often, let's say it caused paralysis of, say, the heart muscle, that means that someone can die. Okay, so without venom inhibitors, these toxic proteins can disrupt cell function. Now, what venom inhibitors do, which are these little molecules in our blood, they bind with the proteins, so with those toxic proteins found in venoms, to prevent the proteins from doing whatever it is they would do to body cells. So let's look at the same example now with venom inhibitors, and you can see these little blue molecules have bound to the active site of the toxic protein, and by doing that, this protein can't attach here to the channel protein. Okay, so it can't disrupt cell function, and these ions are still able to move into the cell as they should. Okay, that's all you need to know. The key point is that the venom inhibitors bind with the proteins found in the venom. Okay, so they're not technically acting on any of our cells. They're kind of stopping the venom, which in this case would be the pathogen that is going to cause disease. Okay, so just jump into question 3. Okay, we're going to look at our two responses now. We've looked at some of those molecules. Two really important responses as part of our immune system. And you've probably experienced both of these at some point in your life. So the first one is fever. So fever starts when macrophages, so remember they are our phagocytes, they're really important leukocytes, release cytokines, so signaling molecules that are called interleukin. So interleukin is a type of cytokine. Okay? Interleukin sends a message to the hypothalamus, which is in the brain, and it's the part of the brain that's responsible for regulating a lot of our body functions and our endocrine system. It sends a message to the hypothalamus to increase the body temperature. Okay, so if you look at this picture, we had our pathogen here. The macrophage, when it engulfed that pathogen, released these cytokines called interleukin, and they will act upon the brain. And the brain has increased the internal body temperature to around 38 to 39 degrees Celsius. Okay, so remember our normal body temperature is around 37.5 degrees Celsius. Okay, so it brings the whole body temperature up. This is a protective thing. So fever, we often associate fever with being sick, and I think a lot of people think that fever is caused by certain pathogens. It's not. Fever is your body trying to protect you, okay? And it's doing this in two main ways. One is that by increasing that body temperature, the body temperature goes higher than the optimal temperature for the pathogen. Okay, so pathogens that survive well in humans like 37.5 degrees. Remember, we look at the impact of temperature on things. If it goes even slightly higher than that, let's say up to 38 or 39 degrees, that could be the critical temperature for the enzymes inside that pathogen, and it will stop working. Okay, so one way that fever helps is that it might make the temperature too hot for the pathogen, and the pathogen will die. Okay, and it goes, within this this kind of variation that humans can survive, but we don't want to be in a high temperature for too long. And if it goes too high, people can actually die from fever if you're going up kind of 40 degrees. The other way it does that is that it promotes the person to rest. Okay, if anyone's ever had a fever, you feel tired, you feel achy, you often get a headache, you get these hot and cold chills. You're not going to get out of bed, you're not going to go around and spread the disease, but also you're giving your body an opportunity to repair itself. An important thing to note with fever is that this is a body-wide response. And what we mean by that is your entire body temperature will go up. It's not just in that one area. So let's say you had a lung infection. The body doesn't just increase the temperature of the lungs, your whole body temperature will go up. Okay, so go in and answer question 4 about fever. And then we have another response called the inflammatory response, or inflammation. And there's a few key points to this. It's really important that you know it. This always comes up in the exam. So the purpose of the inflammatory response is to bring leukocytes and non-cellular components to the area of infection. So all those things that fight off the infection, we need to get them to the infection. Okay, and a really key difference with fever is that this is a local response. And we're going to use the example here of someone cutting their finger on, say, a rusty nail. Okay, so what we mean by a local response is I've cut the index finger on my left hand. That is where this response will happen. It's not going to happen in my right hand. It's not going to happen in my nose. It's going to happen where that pathogen is. Okay. The three main aspects of the inflammatory response. So firstly, we get initiation, so the start of the response. So the cells that get damaged, so let's say we've got a rusty pin here, and this here is the skin. Okay, we've got here a rusty nail, and then we've got this bacteria on the outer surface of that pin. So when the skin gets broken, this pathogen has breached the barrier and is in the body. Okay, the damaged cells will release cytokines, so these kind of chemical signals here, to attract immune cells. And remember, we looked at mast cells. They're a specific type of cell. We have these mast cells, and they release a molecule called histamine, which we mentioned before. Okay, so histamine travels to the blood vessels, and what it does is it causes the blood vessels to open up, so to become wider, and to become more permeable. So remember, something that is permeable lets things go in and out. Okay, we call this process vasodilation. You should have looked at this in year 11 when you looked at regulation of body temperature. Vasodilation is basically making blood vessels bigger. So if you look, this was initiation. Here we've got the vasodilation. So this blood vessel has widened, and it's also kind of become permeable. It's letting things go in and out. And that's really important because these are our white blood cells and we need them over where the infection is. Okay, so the third thing is called migration, and that's basically when all of these leukocytes and non-cellular components that we need at the area of infection migrate to that area. And we can see that happening here with these arrows. We've got our white blood cells are moving up to where the pathogen is, and that's to try and destroy that. Depending on what the component is, it will be doing a different thing. So phagocytes, for example, are going to go around and consume the pathogens as they do. Our complement proteins, they might be doing things like coating that bacteria so that the phagocytes can see it. They might be lysing the bacteria. So remember in a couple of slides ago, the different things that complement proteins do. And also platelets. So platelets are kind of blood materials, and their job is to form blood clots. And this is really, really important because what a blood clot does is basically seals up this open wound so that no more bacteria can get in. Okay, and that's where you get scabs when you have damage to your skin. The blood clot is really important to stop other things getting into your body. Okay, with the inflammatory response, I think it's a really good idea to try and relate this to if you've ever had inflammation, what it feels like. Okay, so hopefully you're thinking things like, well, it's often a bit sore, it's a bit swollen, it might be very red and warm. Okay, all of these things are because there's more blood going there. Okay, this could be if you've rolled your ankle, the same process will happen. You get swelling, you get redness, you get warmth. If you've got tonsillitis, you're actually getting the same thing happening in your throat. If you are someone that gets allergies, often this histamine is causing an inflammatory response in your nasal passages. Okay, so try and relate it to what you know inflammation feels like, and that will remind you, oh, okay, it's about getting increased blood flow. So I need blood vessels to open up and become more permeable so I can get all these white blood cells to the area. Okay, So once you've got all of that written down, I really recommend either drawing a picture or getting one from the internet for this. Jump into questions 5 and 6. It's worth looking up a few different diagrams of inflammation. There's so many different representations of it. So it's good to be familiar with a few different ones. So same kind of thing here. We've got the broken skin here. We've got our pathogens. They stimulate the mast cells to release histamine. So our histamine would travel to the blood vessels. Blood vessels open up. We get our white blood cells leaving the blood and heading to the area of infection. Okay, so worth looking at a few different pictures. Okay, so we've done barriers. We've looked at the role of phagocytes and other cells. We have looked at today what interferons, complement proteins, and venom inhibitors do. And we've gone through these two really important processes of fever and the inflammatory response. Okay, I know there's a fair bit to that. It's quite a lot of information in there. So you really should be coming back and having a look at this and trying to connect, things like what's the relationship between apoptosis and phagocytosis, or what is the relationship between phagocytes and antigen presenting cells. Starting to try and make those connections. And the next thing we'll move on to once everyone is confident with the innate response is looking at this specific response. Thank you.